Hi guys, my name is Sarah. I'm one of the dermatology assistants here at South Shore Dermatology, but what you might not know is I recently graduated with a degree in biochemistry, so I wanted to hop on here and answer some really commonly asked questions about mRNA vaccines, particularly those developed by Pfizer and Moderna to help combat the coronavirus pandemic. So your traditional vaccine, something you might get at your doctor's office every year for the flu, works by injecting a sort of dead virus into your cells. And so in this way, your body develops a sort of memory for this virus, and we like to call that memory antibodies. So the next time you're at the supermarket and someone coughs on you, you don't have to worry as much about getting sick, because if you encounter that virus again, your body has a good memory for it, and it can mount the proper response and stop you from getting sick. Now, mRNA vaccines are actually really similar. The only difference is mRNA is injected, and you can think about mRNA as a sort of blueprint for a protein. In the case of the coronavirus vaccine, the protein in question is the spike protein of the coronavirus. Basically, your cell uses its own machinery to translate it into the spike protein, which isn't enough to get you sick, but it's enough to do that same kind of thing as the dead virus, develop a memory against it. So to explain a little bit about how this mRNA vaccine works, I have to do a little bit of a biology lesson here. So all of our cells contain something called DNA, and DNA codes for all of our genes in our body, every single protein, basically everything that keeps us alive. And so in order to be turned into proteins, the DNA first has to be transcribed into RNA. Once it's transcribed into RNA, it can then be translated into proteins, and then it can go about its business. It can do whatever it needs to do. So when this vaccine is given, mRNA, it takes up the same exact machinery that we have in our cells, that's naturally occurring, and it creates that spike protein I was talking about. Now, the important thing about this process is it's a one-way street. We go from DNA to RNA to protein. There's really no way for the RNA being injected to alter your DNA in any way, cause any other adverse effects, and that's because RNA is also, it doesn't live very long in the cell on its own. So it's sort of translated immediately, and anything that's left over gets degraded. Another question I get asked frequently is, how did we develop this vaccine so quickly? And the answer is really twofold. The first part is that mRNA vaccines have been something that's new to the public, but scientists have been excited about it and working on it for a really long time now. And the other reason that these vaccines were able to be developed so quickly was the high prevalence of the disease in the population. In plain terms, this means a lot of people were getting sick with the virus in a short period of time. This means people who were in clinical trials were infected at really high rates, so we were quickly able to see the efficacy at not only not contracting the virus, but not getting severely sick from it. And we were also able to see in this large study that there weren't any real severe side effects from getting the vaccine. And that's how we were able to develop it and approve it so quickly. Thanks guys for all asking such great questions about mRNA vaccines. I hope I helped answer some of them today. If you have any other questions, please feel free to ask us at the office, and I do truly hope that you get vaccinated. It's one small thing you can do to ensure we all get back to life as we knew it.